Today's class we will be discussing about basic cell biology and the structure of a cell and the constituents of a cell, and most importantly the cell injury, how a cell behaves in case of an injury, and how cell adapts to different conditions. Say for example, we all know why we need to study the basic cell biology and the cell injury mechanisms. Say for example, we have myocardial infarction. Okay, so myocardial infarction we all know is heart attack. What happens when there is a heart attack? Okay, let's say a heart is a 4-walled chamber and we have 3 main arteries that are supplying blood to the heart, and there is a block here, and the area supplied by this artery is not getting enough food and oxygen. So what happens? So this leads to ischemia, right? Ischemia means low blood. Okay, so this ischemia is, it implies 2 things. It leads to 2 things. One is like decreased oxygen supply, or also called hypoxia. Okay, and it also leads to lack of nutrients. Okay, so it could be hypoglycemia, whatever. Basically, it is not getting enough food. So this area, because its demand, metabolic demand is very high, it gets the cell gets injured because of hypoxia and lack of nutrient, and the cells, what they do, what happens to them? They die. So when they are dying, that means what? The injury that is happening here is irreversible. Okay, so this is irreversible, and once it is dead, okay, so what happens after this? So there is this death is called necrosis, and this localized necrosis is what we call as myocardial infarction. Okay, so this is one kind of injury. So the injurious agent here is hypoxia and lack of nutrients. So in the similar way, if you look at a liver, okay, if you look at a liver, and we see infiltration of fat in the liver cells, we call this condition as fatty liver. So here, because of the injury, we don't know what is the causative factor here. It could be alcohol, it could be excess fat, or it could be metabolic derangement in the liver cell. We don't know, but finally there is fatty liver. So what kind of metabolical abnormality that has happened? This we need to understand. If you know the underlying mechanism, what are the different kinds of injurious agents, what is the pathogenesis of this particular condition? Say for example, we'll take one more example and we'll start with the class. So this is a blood vessel, right? When we are planning a diet for either hypertension or diabetes, so what are our what are our principles? Hypertension, low salt diet, salt restriction diet. That's it. Rest of the calories are balanced, and that's what we do. And when it comes to diabetes, the same thing. But we need to understand what is this sugar, excess sugar, doing to the blood vessel. What are the reactions? Why this sugar is toxic? What kind of reactions are happening inside? The cells that are exposed to this excess glucose. We all know, we discussed this in detail in diabetes class. Please go through that again. That excess sugar in our blood leads to formation of advanced glycated end products, right? This results in formation of reactive oxygen species, and they act on the endothelial cells of the blood vessel or the capillary, and finally result in hyaline formation of the blood vessel in either it is both in kidney as well as in other arteries, peripheral arteries. So this is the mechanism. So in this class, what we do is we have a basic understanding of what is the structure of a cell like, and what are different cellular injuries and how the cell reacts to that, both to reversible injury, what are the different morphological features that we'll see, and in irreversible damage also. So we'll see apoptosis, necrosis and all this in this particular class. Okay, so our topic would be like cell injury and cellular adaptations. Okay, now we'll have a look at the structure of a cell. Okay, a basic human function cell. So what is a cell? A cell is a basic functional unit of human body. Yes, a basic functional unit of human body. It has got a cell membrane and a nucleus, and in this what we have chromatin and nucleolus, right? And what else? What are the most important, one of the most important cell organelles? These are mitochondria, right? Mitochondria are also called what? Powerhouses of the cell. And then we have what is known as Golgi apparatus. We have what is known as a Golgi apparatus. And then of course we have this endoplasmic reticulum, both smooth and rough endoplasmic reticulum, right? And we have what is known as lysosomes, usually seen at the end of this Golgi apparatus. And what else do we see? Nucleus, mitochondria, endoplasmic reticulum, smooth end, this thing, Golgi apparatus. Yeah, basically, and we see some kind of centrosomes. So what are they? Centrosomes. So centrosomes are the places where you have microtubules and thin filaments inside called as microfilaments. Okay, now we have the cytoplasm. Okay, so this is a cross section of a cell. Now if you look at the cell, one of the most important organs in a cell is, yes, not nucleus, it is the cell membrane. Okay, the moment the cell membrane is damaged, the cell death is very quick. So cell membrane is the most important part of the cell. Now if you look at the cell, the plasma membrane, the structure of plasma membrane is something like this. It is made up of basically fat, phospholipids. So it has a double membrane, phospholipid, okay, and it has got a head and a tail. So the head part here is hydrophilic. I'm sorry. Yeah. Hydrophilic. That means what? Hydrophily means it attracts water, and the same is with internal. So this is the external layer, the external facing. This is inside facing. So we take a small cross section of it. This is external and this is internal. So the lipid, the phospholipid is basically lipid. It is both phospholipid plus phospholipid is both phosphorus plus lipid. So this is a phospholipid layer interspersed with what do we say, the proteins. Okay, so basically the structure of plasma membrane is made up of both lipids and proteins. The lipid in plasma membrane is called phospholipid and the proteins are basically transmembranous. So if you look at this protein and try to see the structure here, it will be something like this. Okay, this small unit is communicating from the external world to the internal world, and some of these proteins, they also contain some kind of carbohydrates. So carbohydrates, what is the importance of these carbohydrates? Say for example, it is more like an address, right? So you see houses and you see a nameplate saying we are so and so. So in that way, the external environment is composed of what? Basically blood. Every cell is supplied by a capillary, is it not? Is supplied by a capillary, and there are so many antibodies and other proteins that are moving in this. When they come to this home and try to identify who you are, these are more like nameplates. These carbohydrates are more like nameplates. So through these carbohydrates, basically our immune system identifies whether this cell is foreign or innate. So this is the structure of this plasma membrane. What is the function of this protein? These proteins, they do different things. They serve as like transporter molecules. Say for example, if one of the items need to go in there, say for example, if this is hydrophilic, right, the membrane is hydrophilic, so fat, say for example, you need to take glucose, for example, you need to send glucose from external environment to internal environment. So these proteins, they work as glucose transporter molecules, and they also work as sodium potassium ATPase. Okay, this is an enzyme. So sodium potassium pumps. So using ATP through this enzyme, sodium and potassium. We all know that sodium is highly concentrated in the extracellular space, whereas potassium is high in this intracellular space. So these proteins, the membrane proteins, what they do is they facilitate the movement of ions, nutrients and other kinds of hormones. They work as receptors. They also work as receptors, right, to different kinds of hormones and different kinds of neuropeptides. Yeah. Next to plasma membrane, one of the most important organelles in our body is nucleus. So nucleus has also got its own membrane. It's also a bilayer membrane, just like plasma membrane, and it consists of nucleolus. This is nucleolus, and this is called chromatin, right? So chromatin is loosely clumped DNA protein. So why we are calling this chromatin but not as chromosome? See, if the cell is not in a divided state, when it is not dividing, all the genetic material, they clump together and we call this as chromatin. But when the cell is dividing, is becoming two parts, and these form 23 pairs of chromosomes each, and they divide. Only chromosomes we can see only at the time of division. Okay, so what is the function of nucleus? Nucleus function is basically to make proteins. So the DNA that is there in the nucleus codes for specific protein. So it contains RNA, ribonucleic acid, DNA, ribosomes, and some kinds of proteins. And the nucleolus here is exclusively codes for some of the ribosomes that are there in the cells. So the ribosomes are essential for making proteins. Okay, so this is about nucleus, and next comes our ribosomes. So ribosomes are like sausage-shaped structures. Basically, they are also called powerhouses of the cell. So whatever nutrients that are coming into the cell, whether it is carbohydrate or protein or fat, so from that, if energy is to be released for sustaining of this cell, that energy release in the form of ATP, adenosine triphosphate. So this is also called cellular currency. Okay, cellular currency. One glucose molecule enters from blood into the cell, and that gives finally a net of around 36 ATPs. Okay, so this is what this mitochondria does. And what else do we have? Yes, we have endoplasmic reticulum. Endoplasmic reticulum are like loosely connected channels. Okay, so there are two types. This is smooth endoplasmic reticulum. This is rough endoplasmic reticulum. So the roughness is because of the presence of these small tiny dot-like structures called ribosomes. Okay, so this rough endoplasmic reticulum, what they do is they help in the synthesis of protein, and the smooth endoplasmic reticulum, they synthesize these steroids and lipids, basically phospholipids that are necessary for maintenance of the integrity of the cell membrane. Okay, and finally, what else do we have? We have this Golgi apparatus. Golgi apparatus, what it does is, once the proteins are coded for by the nucleus and they come out, all these proteins are stored and packed inside this Golgi apparatus. So this Golgi apparatus, when the time comes and fuses with the cell membrane and releases these proteins. So Golgi apparatus is basically a storage for all the proteins that are produced by a particular cell. So these are basically the main important cell organelle in a cell. But there are other kinds of few more cell organelle. So we can call it as connective tissue of the cell. Okay. So the connective tissue of the body is to give the shape and the flexibility or the form of the organ, whereas the connective tissue inside, because this is a cell, they are called microfilaments. Microfilaments, they give form and shape to the cell. And this centrosome, where is this? The centrosome, what it does is, it contains microtubules. Right? So, what are microtubules? Microtubules are a bit stronger than these microfilaments, but what do they do is, they give different kinds of shapes to the cell. Say for example, if you take this, digestive system, okay? We all know the intestine contains microvilli, is it not? If you take the structure of, let's say, a small intestine like this, okay? So this is a digestive system from here, duodenum, and finally it ends up in small intestine. Now, for absorption, you need to have more surface area. Now, if this is like this, so it has got a small surface area. How do we increase the surface area without increasing actually the length of intestine? If we push this inside like this, right? If we push this inside and again like this. So in the same space the food is coming in contact with more surface area, is it not? Okay. So these kind of structures from the underlying cells, let's say this is an enterocyte and this is another enterocyte, and we have another enterocyte here, another enterocyte here. So these projections are formed by these microtubules. The movement for phagocytosis, say for example, if you see a WBC, right, and here is our bacteria. What it does is it increases. We know, I mean, we have seen it in amoeba and all this stuff, that it comes and connects and ingests this bacteria. For that, the microtubules are important. So basically, this is the gross description of cell and its important cell organelle. So now, what is the function of a cell? When we say cell is a basic functional unit and an independent functional unit, okay, so we all know our body is made up of about 50 to 100 trillion cells, but that means it is a colony of 50 trillion cells, but each cell is an independent functional unit. So oxygen is coming in and carbon dioxide is moving out. That means the cell is capable of doing its own respiration, is it not? In the same way, it is taking in protein, metabolizes and sending out urea as a byproduct. So it is able to do its own excretion, elimination of waste products, right? It is able to replicate on its own. So it is able to do its own reproduction, right? And it is taking food, right? It is taking what? Carbohydrate and releasing its energy. So basically the cell is a functional unit on its own. I mean the functional unit of a cell, but it is a self-sustaining unit of its own. Now when we understand the mechanisms that underlie different conditions, okay, how a cell behaves in regards to different kinds of injury. It could be a physical injury, it could be physical in the sense, it could be a thermal, that is heat, radiation, or like changes in atmospheric pressure, or brute injury, physical injury as such, so by physical agents, or it could be hypoxia. Say for example, this particular cell, okay, we discussed already that every cell has got a capillary supplying food and oxygen. So now if the oxygen is not going inside, that is a hypoxic injury, right? It is not getting its oxygen. That can be fatal to the cell. And what else? What else? The physical crush injury. I mean, the cell membrane is damaged because of, let's say, heat or chemical or something or that. So that is physical agent. And what else? Infection. A bacteria comes in and attacks the cell. That is infection. And what else? We have nutritional deficiencies. Say for example, you are not giving enough protein, right? No enough amino acids to make the protein. So the cell cannot make its own proteins, or there is no enough fatty acids. So it cannot make its own lipid membrane. And what else? Could be some kind of chemicals. Yes? Some chemicals may come and interact. Say for example, what are we consuming when we are taking a soft drink? We are consuming so many additives, coloring agents, stabilizing agents, all this stuff. So when they come and interact with the cell, they may lead to some kind of damage. That is also an etiological factor. And what else? What are other kinds of injuries that can happen? Psychogenic, right? stress, when these cells are exposed to a lot of stress hormones are released, that itself can be an injury. And some kinds of drugs, say for example, non-steroidal anti-inflammatory drugs, also called painkillers, they are harmful to the liver cells. Okay, and what else? Iatrogenic. Iatrogenic is caused unintentionally by a physician in a hospital. That is iatrogenic. And what else? Idiopathic. Sometimes we don't know what is the cause. Right? Say for example, essential hypertension. What is essential hypertension or primary hypertension? Primary hypertension is the hypertension for which there is no identifiable cause, but still there is a disease, an injury to the arterioles, and that results in increase in hypertension. So we have to, since we have covered cell, we have to look into the cell injury. And in cell injury, first thing is like etiological factors or causative factors. Causative factors includes hypoxic injury or physical injury are caused by chemicals, caused by infections, nutritional deficiencies, psychogenic, drugs, iatrogenic and idiopathic. Okay, so these are some of the etiological factors that are responsible for cell injury. So now we see what are the underlying mechanisms that are there in a cell. Okay, so one point we need to understand here is that the cell currency, the energy currency for our cell is ATP. So for production of ATP we need what? Oxygen. So hypoxic injury is one of the most serious injuries that our body can sustain. Now we see what is a hypoxic injury, what is the underlying mechanisms, what will happen, what are the reversible cell injury features and what are the irreversible cell injury morphological features. We discussed cell and an introduction to cell injury, right? So when it comes to cell injury, we saw some etiological factors. What are they? We'll just recap once. That one is ischemia, and the second one is physical agents, and the third one is like chemical injury, and fourth one, let's say immunological or immunology mediated. Fifth one is nutrition, and sixth one is like iatrogenic, that is physician caused. Seventh one, let's say psychogenic, and eighth one, what else we have? Iatrogenic, psychogenic, idiopathic. So like this, there are so many etiological causes for cell injury. Now, most importantly, we'll see what is ischemia and how it causes injury. Okay, so there are two things we need to study when we study cell injury. One is like what is the underlying mechanism through which the injury is being addressed. What are the morphological changes in the target tissue? Okay, so underlying mechanism, let's say it could be free radical damage, right? If the target tissue is cartilage, and the cartilage, if it is inflamed, it may lead to arthritis. Okay, this is morphological changes in the target tissue. And what is the underlying mechanism, how a cell responds to an injury. So this is how we have to look into. So hypoxia, ischemia means, what is ischemia? Ischemia means, let's say there are groups of cells here and the blood is coming through capillary, right? Let's say this capillary, Okay, the blood is coming to this area and the blood vessels, what are they doing? The blood is coming from this arteriole and into the capillary. From there to venous end, it is coming out. Right? Right? So, the blood is coming and supplying food and oxygen through this area. Say for example, there is hypoxia or ischemia. Ischemia means what? There is a block over here. The blood is not coming clearly. So, the oxygen is not coming. So, this results in what? Hypoxia. This results in hypoxia. So, what will happen? This block can be temporary or this block can be a permanent one. If this block is temporary in the sense the blockage is temporary, maybe some alternate routes it gets again blood, or maybe we clear the block in meantime before the cell gets irreversible damage. So the point here is there can be two types of damage. One is reversible damage and second one is irreversible. So the damage because of hypoxia due to ischemia. Ischemia is lack of blood resulting in lack of enough oxygen for the cells in the tissue. This can lead to two types of cell damage, reversible cell damage and irreversible cell damage. So what are the mechanisms that are there in irreversible cell damage? So why do we need oxygen? We need oxygen to break down glucose and rest of the nutrients to release energy and store in the form of ATP. So these cells, they have to create their energy currency and use it for different functions. So now when the oxygen is not available, the production of ATP will come down. The production of ATP will come down. What happens when the production of ATP is coming down? So there is no energy for the cells. So then what happens? Production of ATP is higher with what kind of respiration? Aerobic respiration. Right? So when this fails, what is the other thing for our body? Exactly, anaerobic. So the tissue shifts into anaerobic respiration. Though the ATP production is less when compared to aerobic respiration, still the tissue is able to sustain its life. But when the cell is producing ATP through anaerobic respiration, what happens in the tissue? It produces what? Anaerobic respiration produces what? Lactic acid. So when a lot of lactic acid is produced and it is entering into the blood, lactic acid, what happens to the pH? pH falls. That means blood is becoming acidic. What do we call this condition as? Lactic acidosis. Right? When there is hypoxia caused by ischemia in a tissue, the reversible cell injury, not death, reversible cell injury. Okay, so the first mechanism is, the first one is decreased ATP production, prompting the tissue to go for anaerobic metabolism, and still it is able to produce ATP, but the problem here is it is producing lactic acid and releasing into the capillaries, which is going into the blood, leading to the second mechanism, that is lactic acidosis. What is problem with this? Lactic acidosis. So if this is not happening, we need what is the function of a cell? Cell's function basically is to produce lots and lots of protein. Okay, so and not only protein, it has to produce certain kinds of lipids also. So the plasma membrane protein, I mean, we discussed that plasma membrane structure is like something like this: middle hydrophobic phospholipids on either side, and in between dispersed are the protein. So what happens is the phospholipid production, this comes down. The plasma protein, the protein production will come down. If you look at the ribosomes, what happens is they get dispersed throughout the cell. They are no more attached to any kind of endoplasmic reticulum or any other stuff. So when this is the third underlying mechanism, these are the changes that are happening when a tissue is exposed to hypoxia. And next one is what happens, the protein production and the membrane proteins, they get damaged. When the protein production is decreased, what happens is the membrane proteins, they gets deregulated. I mean, in other sense, they lose its functionality. They lose their functionality. Functionality of these membrane proteins will be decreased. So that what will happen, when the membranes they are not tight enough, the water will move into the cell. Okay, we all know that plasma membranes are semi-permeable membranes. They selectively allow certain things. They maintain a gradient. Okay, extracellular fluid is more of sodium and calcium. Intracellular is more of potassium and magnesium. So when the plasma proteins are not strong enough, the cells are not able to make sufficient proteins, and the ribosomes are dispersed, what happens? The membranes, they become leaky. So they become swollen, right? So these are the underlying mechanisms in irreversible cell damage. One is decreased ATP, second one is acidosis, third one is decreased protein synthesis and membrane protein leakage. So these are the damages that occur in reversible cell injury because of hypoxia. Okay, just a recap, because this is very, very important. What happens is, once we know the cellular mechanisms underlying behind any kind of disease, so what it tells us is, it gives us an opportunity. Most of us, we believe that once there is a hypertension, this is a permanent. But the changes that are happening in artery, or in case of diabetes, the changes that are happening in an arteriole are just reversible cell injury type. They are not permanent. That gives us an opportunity to look into the cause of injury, and from there we can work on. So I request all of you to go through these classes again and again, so that you get the concept right. So initially it may look overwhelming, but give yourself some time and try to get the concept. Once the concept is clear, it is very easy to deal with our clients or customers or patients very confidently. Now, if this situation continues, that the hypoxia still continues, the pH rises, and there is decreased production of ATP, so the membrane proteins are not strong enough, and the functionality of the membrane proteins is lost. So what happens if it still continues? They become so leaky, it leads to irreversible cell injury. And irreversible cell injury means also the cell death. Right? What is the underlying mechanism? How a cell dies when there is no oxygen? So what happens is, the first thing is, there is, as I said, the extracellular calcium is more compared to intracellular calcium. So the calcium influx happens. That means calcium, this is all extracellular fluid, is it not? The capillary blood. And this is intracellular fluid. From here the calcium comes in and damages mitochondria. Okay? So the first one is because of calcium influx, what happens? The mitochondria, they gets damaged. So there is absolute stoppage of production of the ATP here. The second thing, what happens is, how a cell dies. So what is the most important organ of a cell? Its cell membrane. If once its integrity is lost, right? So integrity of the cell membrane is maintained by what? The phospholipids. So this increased calcium, it increases the activity of lipases. So they destroy the cell membrane. So the cell membrane becomes, right, like this, leaky. It's all protein, all phospholipids are gone, right? So when this happens, the water comes in, it swells up, and it leads to increased activity of proteases, which form the microfilaments. We have seen the structure of cell is maintained by microfilaments, which are made up of proteins. So now the pillars are broken down. So microfilaments, the proteases, they are digesting the protein. And finally, so the wall is breached, the pillars are broken down, and they are reaching the core, the nucleus. So the nucleus is digested by endonucleases. What they do is, they rip apart the DNA. And finally, once this is done, what should happen? Whatever protein core that is there is digested by hydrolyzing enzymes. Hydrolysis. So this is the underlying mechanism. So once this is done, everything is damaged. Where is it going? It is going into, till now it is an integral structure. Now it is broken down, and so many things are released. Breaking of phospholipids, the end products are released into the blood. Breaking of these endonucleases, the end products are released into the system. Okay, so we see certain enzymes in certain kinds of diseases. Say for example, in heart we see certain kinases. So we see CPK, we see. So when these things are broken down, the enzymes, they give an indication that the damage, there was an irreversible damage because of breakdown of the cell completely. Okay, just to recap, irreversible cell injury, which means cell death, occurs following the leakage of calcium into the cell, which activates lipases. That means, see how a structure is collapsed. First we break the walls. So the lipases, that means the lipoproteins that are making the phospholipids that are making the cell membrane, are broken down. Next, the big pillars, cytoskeletons and all, they are broken down, and finally they come into the core, that is the nucleus. They are digested, and rest of the things are hydrolyzed, completely washed out, broken down. So when this happens, we call this as irreversible cell injury or cell death. Now we have to see what are the morphological changes, what else are the other kinds of what kind of morphology an organ have when there is irreversible cell death. Again, it depends upon the organ, the target organ where the cell death occurs. It differs from skin to heart to spleen to liver. For every organ, the morphology differs. So we look at some of the morphological features that happen in irreversible cell injury, otherwise known as cell death. Okay, so before going to that, I would like to tell about one interesting condition, that is damage mediated by free radicals. Okay, so when we talk about free radicals, right, first of all, free radicals are also known as reactive oxygen species. Okay, what are these reactive oxygen species? So if you look at oxygen is 8. Okay, oxygen, you have 8 protons, and so obviously we should be having how many? 8 electrons, right? Oxygen is molecular atomic number is 8. That means the number of protons are 8. So number of electrons also should be 8. Okay. Oxygen has got 2 unpaired electrons. So if we go back to our atomic structure and see, so if this is nucleus, so we have 1s orbital. That means first orbital contains 2 electrons. Then the second orbital contains 2s and 2p. That means 1s orbital contains 2 electrons, and so now we have 1, 2, 3, 4. We have to accommodate 3 more electrons. So p, 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 and we have only 1 is paired in p. So 2 electrons are not paired. Okay. So in other words, an oxygen molecule contains 2 unpaired electrons, and it reacts with another oxygen molecule with 2 unpaired electrons and forms a double bond, forming oxygen molecule. Got it? Now the same thing, same oxygen which contains 2 unpaired electrons react with 2 hydrogen molecules with 1 electron each, and they form a single bond forming H2O. So this is a stable compound, this is a stable compound. Now what happens is, in our body, oxygen is the end receiver of electrons, right? Electrons, it is the end receiver of electrons. So if you look at glycogen or glucose metabolism, we have what? 3 stages. Glycolysis, where glucose is broken down. Second thing is Krebs cycle, and the last one is electron transport. Okay, during electron transport, the protons are pumped out of mitochondria and the electrons are pumped in, and oxygen is the one which collects the electrons along with 4 electrons plus 2 hydrogen molecules forming water. So we'll discuss this in detail in biochemistry class, but for time being, during metabolism, this water is formed, okay, and this free oxygen is the receiver of electrons. Okay, we need to understand. Okay, let me recap. What happens is, during metabolism, glucose releases energy. In this process, during the final stage of electron transportation, oxygen is the one which receives electrons from this electron transport proteins in the mitochondria. If there is an impaired metabolism in the sense, so the metabolism is not happening in a proper way. So this results in some imbalanced molecules, species of oxygen and its derivatives. So they are hydrogen peroxide and hydroxyl and superoxide. So superoxide is having 1 unpaired electron, hydroxyl is having 1 unpaired electron, hydrogen peroxide is having unpaired electrons. So what do? These are formed due to impaired metabolism in our cells. So what happens is, these free radicals, because they are unstable, they go and attack molecules of each and every constituent that is there in the cell. So in a sense, what they are doing? They are oxidizing. So what is oxidizing? What is oxidation? If you let iron to react with oxygen, it forms iron oxide. Okay. So What happens here? Iron oxide is formed, and this is nothing but rust. So rusting is nothing but oxidation. So this oxidation of cell happens because of this production of free radicals. So these free radicals are one of the causes of cell injury and resulting in cell death.